Thank you very much, and good morning to those of you who are here. And I think we'll start filling out this auditorium as we speak. Following disclosures. So I'm going to talk about a few aspects of prostate cancer that will obviously involve genomics, but I wanted to start off with a little background, as this is an educational session, with regards to the epidemiology of prostate cancer, which actually plays an important role in what prostate cancer looks like, as well as talking about germline and then a recent genomic understanding of prostate cancer. And I think it's a good way to start to think about prostate cancer as two diseases. Charles Dickens, probably one of the most famous opening sentences in English literature, it was the best of times, it was the worst of times, thinking about London and Paris in the time of the French Revolution. And this is really also a good way of thinking about prostate cancer, where in the early disease we have a very different question than in late disease, and I'll describe that. This is the first time that I've ever been in a session where prostate cancer has been considered a rare disease. Worldwide, there are around 900,000 new cases per year, with a fairly large number of deaths. It's the second most common cause of cancer in men. In the United States, the statistics show that it's the most commonly diagnosed cancer and the second most common cause of death, cancer death. And if you look at 5-year survivals for patients with prostate cancer, and I'm just comparing it to breast and lung cancer, you'll see that in the early disease, clinically localized disease, virtually you have almost 100% 5-year survival. In distant disease, it's a lower percentage, but overall there's a very high percentage, and that really reflects the differences between the early detection of prostate cancer through various testing and late disease. So what are the reasons for this? And this just shows what happened in approximately the early 1990s when PSA screening, so a molecular test, was introduced, and large-scale screening identified many prostate cancers. There was a peak of prostate cancers that were in the population, and this number stayed quite high until this date here, where the number of incident prostate cancers, at least in the United States, dropped. So what's the problem with this molecular test? Won't go into the details of these two very large studies, but a European study and a US study asked the question, does this PSA screening help decrease the mortality of prostate cancer? And in fact, in the European study, it demonstrated a benefit, that is, PSA screening decreased the number of mortalities over a number of years. In the US study, it showed that there was no improvement or use of the PSA screening, and this led to a very important event with a recommendation, at least in the United States, that PSA screening should not be done. Now, I think this is an important lesson because this has impacted not only the epidemiology of prostate cancer, but also how it's being treated in some countries, the United States in particular. And this is some thoughtful reanalysis of the data where when one looked at the control group, that is those men who were not PSA screened, what we see here is that up to 90% of those men had actually undergone PSA screening. So really, in many people's view, this is disqualifying this type of data. But what's the effect? So this change in the way that we think about prostate cancer and PSA screening has led to a decrease in the PSA screening, and importantly, for clinicians who examine patients, there's a dramatic decrease in performing digital rectal exams, which are one way of detecting nodules that could be associated with prostate cancer. And if we look at data from two eras, so in green and in blue, we see that this decrease in number of cases will be quite important, but particularly in older population of men. So what this really tells us is that we have a molecular test. Unfortunately, we probably have the wrong conclusion to certain larger epidemiology tests that told us that we probably shouldn't be screening for PSA. and this has to probably be readdressed. And what is the consequence? So the real-life consequence is, most likely, that we're going to start to see an upsurge, and here's just some initial data showing that we're starting to see increased numbers of men who are coming to the clinic with advanced disease, already metastatic prior to initial diagnosis. And we're also seeing an increase, a slight increase in the change in mortality, where instead of seeing a drop, which we've seen over many years, we're starting to see a leveling off, which is of great concern. So now I want to turn to germline understanding of prostate cancer. And there have been a number of Scandinavian studies looking at twin registries and asking the question, in common cancers, which are the most heritable cancers? And these are just some of them, prostate and melanoma being the most common. These are two studies, one more recent from Dr. Mucci's group. In most of the studies looking at SNPs or much fewer studies looking at copy number variants, the outcome or what the trait that we're usually looking for is the presence or absence of prostate cancer. If one were to do this, one realizes that in most of the studies, there are some rare variants that may be deleterious, and I'll talk about that in a moment. And then there have been some larger GWAS studies that have very common alterations that have very small effect. And this is just a summary from the, I think, a very large consortium study where around 24,000 men were screened for common traits, and the common traits all adding up together were not able to help improve the ability to understand which men would have aggressive disease that led to prostate cancer-specific death. When one looks at less common traits, one had put germline alterations such as in BRCA1 and 2 into this context, and this was thought of as relatively rare. But recent work looking at patients with advanced cancer from Pete Nelson's group and Colin Pritchard leading this group and looking at a number of cohorts of patients with advanced disease have identified that up to 12% of patients with advanced disease actually have germline mutations or alterations in BRCA2, ATM, and other genes associated with germline alterations of DNA repair. Now I'm going to turn to the genomics. These are somatic alterations in prostate cancer, and this will cover both the early and the late disease. And I wanted to start out with just showing a historic view, and I think what's important is that if we look historically, we've known about many of these alterations for a long time, such as 10q loss in somatic, in the tumors at the somatic level, a c-MYC amplification, as well as p53 loss and AR mutations. But it's not till more recently, using more extensive genomics, that we're able to look at this in a context of therapy and in larger numbers of cases. So I'll talk a little bit about that. And obviously, next generation sequencing has helped, but also other technologies that I may mention. Lung cancer, for those of you thinking about cancers that can be very well defined by genomic alterations, really has an established molecular classification. EGFR mutated, ALK altered, KRAS wild type or mutant are very important to know clinically. And that's in part due to the fact that we have drugs now where it makes a difference to know that information. In prostate cancer, we also can define specific genomic subtypes. So for example, around 50% of prostate cancers harbor a gene fusion that involves an ETS transcription fusion, ETS transcription factor. And I think the important, and what's actually quite interesting about this translocation is it puts together an androgen-regulated promoter region, so TMPRSS2 is the promoter, and fuses it with pretty much the full length of an ETS transcription factor. And this can be shown at the DNA level with FISH assays or now by immunohistochemistry. And I think it's a very nice illustration here where we have cancer in brown that shows a translocation. And at the very point where it transitions from a pre-malignant lesion into a cancer, and then we have the benign tissue, you can see that there's no expression. So it appears to be a very specific event in prostate neoplasia. And as I mentioned, usually through the fusion of TMPRSS2 or androgen-driven promoter gene with an S transcription factor. Since the initial description, there have been a number of other S transcription factors and some less common genes that have been associated with gene fusions. And this helps us start to think about prostate cancer, at least 50% being involved with some sort of translocation. If we look at somatic alterations, we can see that they're genes that are associated with copy number gains and losses, and a very common gene that's lost in prostate cancer is P10. P53 is often lost. When we think about genes that are mutated, there are very few recurrent mutations in prostate cancer. One of them is SPOP, it's listed here. And what's informative, I think, about the types of mutations that we see, that they're all located in the MAF domain, the protein binding domain of this gene. When we look at whole genome sequencing of prostate cancer, we see that prostate cancer, like many other cancers now that have undergone whole genome sequencing, we see large structural rearrangements, both intra and interchromosomal alterations. And just from a very simple perspective, if we think about grade of cancer, so in this case we have prostate cancer that has a grading system that's called Gleason grading. And if we go from low grade to high grade, clinically we can demonstrate that this very nicely distinguishes the low grade tumors from the high grade tumors. And now if we look at the correlate going from low grade to high grade, we can see that the genomic copy number changes correspond very nicely with this grading system. Now, if you look at this, it really looks like a fingerprint that we have the same peaks and valleys of genomic alteration that seem to become enriched as we look at higher Gleason scores or higher tumor grades. The other very important aspect about prostate cancer genomics at the somatic level is that if we take a cross-section of a prostate gland and we circle it here, or here it's colored, for the different discrete lesions, and this is based on morphology and also grade, it appears that each tumor, each prostate gland that has cancer probably harbors up to 5 different genomic alterations. This may in part explain why some cancers, probably the minority, undergo very rapid transition to metastatic disease and are lethal, whereas other cancers are more indolent. So this heterogeneity of the tumors that we see somatically may play an important role in determining a clinical outcome. So summarizing some of that genomic data, I would just show you that we can think about prostate cancer predominantly as ETS-altered cancers, so ERG, ETV1, ETV4. There's another larger class called SPOT mutations that have been identified, and then some other common alterations, FOXA1 and IDH1 alterations. So these are mutually exclusive classes, and I think this becomes quite interesting in thinking about that. Probably the reason why this hasn't caught on the same way as in lung cancer to this date is probably due to the fact that there are no important therapeutic implications yet. If we look at prostate cancer that has been treated, so most of what I just described is all observations that have been made in patients who have not been treated. And if we think about patients who are treated, we start to see an emergence of some different alterations and also some loss of these mutual exclusive relationships. I'll just show you, for example, that the ETS rearrangements I described are very commonly seen with P10 alterations, but mutually exclusive from SPOT mutations, except once they undergo therapy. So what happens when you start treating patients? And this is a very nice study led by Nikki Schultz and Ellie Van Allen as part of a Stand Up to Cancer PCF Dream Team study. And in looking at over 1,000 cases of both early and late disease, one can start to see, looking at this diagonal line, tumor mutations that occur more frequently after treatment. And I would just highlight here that you can see the androgen receptor is mutated more frequently after treatment, as are some other alterations such as P53 mutations and RB alterations. So this brings to the question, what are the changes that occur during treatment? And most of these are related to the androgen receptor. And I'll just go over some of the alterations that we see and some of the mechanisms that we believe are related to resistance. I think it's important as background information to remember that in prostate cancer, we've had targeted therapy for a long time, and that's androgen receptor blockade. And this has been known since the 1940s, where experiments that led to Nobel Prize work from Huggins and Hodges demonstrated that by blocking androgen, one can kill prostate cancer tumors, but they almost always recur once you remove the effects of anti-androgen blockade. This has led to a number of drugs, therapeutics that block some area of androgen synthesis or the actual androgen receptor. This is the evolution that can occur for a patient who will eventually die of prostate cancer, and that is they may be diagnosed with localized disease and undergo androgen ablation therapy, but over time they all demonstrate resistance. And this is just an example, a waterfall plot showing some of the early studies where if a patient who is thought to be castration resistant or no longer responsive to anti-androgen therapy was treated with a more potent anti-androgen, we'd show that it could be shown that they had a much greater response to therapy now. But how do tumors get around that, and what are the genomic alterations that we see? And I just highlight three areas that are really being commonly found. One is there's a workaround. That is that the androgen receptor itself undergoes a mutation or alteration. Other pathways such as PTEN activation occurs after androgen ablation. And the last is an indifference. That is that there's entirely different pathways or transdifferentiation that can occur where the tumors themselves are no longer responding, but no longer in any way relevant to the androgen signaling pathway for their proliferation. So here are just some examples of genomic studies that have looked at this phenomena in the setting of cancer treatment. So this is an example looking at cell-free DNA and asking the question, are there androgen receptor mutations or alterations, and demonstrating that in patients who have these alterations after treatment, they're more likely to die more rapidly of prostate cancer. There's an alternate splicing that can occur in the androgen receptor, and in this V7 variant, the androgen receptor no longer has the ligand binding domain, and therefore, in theory, the androgen deprivation therapy that's targeting that domain is no longer effective. And in a number of studies where one has characterized the splice variant's presence or absence, it appears that there tends to be a decrease in responsiveness to anti-androgen therapy, leading to more rapid death due to prostate cancer in these patients. And just yesterday, or day before yesterday, in Cell, there's another mechanism that was described, and that was alterations in the enhancer, so a non-coding region of the gene that become amplified and that increase the likelihood that AR can maintain signaling and tumor cells can continue to proliferate even in the face of anti-androgen therapy. As I mentioned, other pathways can be activated. So here's just an example of two studies that demonstrated that when you turn off the androgen receptor, you can get activation of the PI3 kinase pathway. And the last area is a transdifferentiation or lineage plasticity. And here's an adenocarcinoma that after undergoing anti-androgen therapy now looks like another cancer, which is called a small cell cancer, which is defined by these small cells, a very little cytoplasm. These are extremely aggressive cancers, and so patients usually die within 7 months' time. And when we look under the microscope, we can see that often these dark blue cells are seen in close proximity with adenocarcinoma, suggesting that they may arise from the same cell. when we think back of the fingerprints of the localized tumors that I showed you before, and we now superimpose the adenocarcinomas and these neuroendocrine tumors, we can see that they almost have the same genomic fingerprints, save a few genes, and those would be P53 alterations and RB alterations. And so in thinking about this transdifferentiation process, one can think about whether they're coming from two different cell types, but the best genomic data and epigenetic data to date suggests that what's really happening is after therapy, we're seeing a transdifferentiation of tumor cells, and we believe that it may be due to epigenetic reprogramming. And there have been some nice studies looking at cell lines that have demonstrated that by knocking down RB and P53, one can at least start to see some of these early neuroendocrine changes, and suggesting a model that's obviously a model in development. It's quite early in the process that a patient who has androgen receptor responsive disease at some point during this blockade may undergo a number of genomic alterations, which could include P53 loss and RB loss, but certainly there could be other alterations. And then through some epigenetic reprogramming may then undergo this transdifferentiation. The importance of this process would be that it would allow for the tumors now to evade the androgen receptor targeted therapy. This is not the only cancer type, so returning to lung cancer, we see the same type of alterations and effect in lung cancer. So it's been well known that in P53 RB mutant lung cancer that have been targeted for EGFR receptor, we also see this transdifferentiation process. The very last thing I just wanted to cover is going back to germline alterations in DNA repair genes. In genomic studies carried out more recently in patients who have been treated, it was observed that there was a high percentage of DNA repair mutations at the somatic level in these tumors. And this led to Johan de Bono's group and others thinking about how some of the responders in their trials might be classified when they look back at the somatic alterations in the patients who responded to PARP in trials that were targeted for patients who may have DNA repair mutations such as BRCA2, BRCA1, ATM. They found that in fact there was an enrichment in DNA repair mutations in the patients who responded to PARP inhibitors. And this led to the study that I mentioned earlier for Colin Pritchard's group and Pete Nelson, where they looked at DNA repair mutations both at the somatic and germline levels in patients with advanced cancer, and as I mentioned earlier, found, depending on the cohort, anywhere from 10 to 20% of patients in these cohorts had DNA repair mutations. These are all potentially targetable and really changes the face of how we think about advanced prostate cancer, and these findings are probably also true for other advanced cancers. And just in last moment, I'd just like to look forward. How are we going to address the genomic sequencing in the future? Clearly, gathering metastatic samples or tissue samples is much more challenging than liquid biopsies. And there's a lot of enthusiasm about either capturing cells and sequencing them, perhaps even more now looking at cell-free DNA and performing genomic analysis on these samples. The major advantage would be that one could potentially identify this major issue of heterogeneity and capture all the information if one can deconvolute the information within the cell-free DNA. So we are at 2018. We're at the point where many thousands of genomes have been sequenced in the area of prostate cancer, both somatic and also germline. And one would think that we've reached sort of the conclusion, but I think in the setting of treatment, we're starting to identify other mutations. And I would just also highlight another Cell paper that came out very, very recently, identifying CDK12 mutations. And these mutations in the study were identified as being a distinct class of prostate cancer, yet another, and that being associated with tandem duplications that in theory could lead to more neoantigens, and then, although I haven't discussed it up to this point, potentially a novel approach for therapy that is immunotherapy in the setting of prostate cancer that up to now has not worked well. So these are encouraging findings that obviously need to be validated. So I won't go through reading all the summary aloud, but just to state that prostate cancer, although extremely common, has some forms that are rare. So I guess that's where we fit into this session. And importantly, in the setting of treatment and resistance, we're starting to see a very different genomic landscape of mutations. Thank you very much for your attention, and I'll end with a sort of peaceful view from Switzerland.